introduce Lucas Cernusak. So Lucas is did his PhD in plant science at the Australian National University in 2004, and he's currently associate professor in the ecology and zoology group at James Cook University in Cairns, Australia. And his research focuses mainly on plant and understanding environmental and biological controls on the exchange of carbon dioxide and water in plants. He also does some really interesting work on the interpretation of stable isotope signals in organic material. And I've had the pleasure of collaborating with Lucas on some of the work that we've been doing on Australian tropical rainforests and looking at how the traits and how these forests respond to climatic variation. So it's great to have you give this seminar. I appreciate it's a late hour in Australia and you've come all the way into your department to make sure the connection is good. So thanks for making the extra effort for us as well. So over to you, Lucas. You're muted at the moment, Lucas. You're still muted at the moment. Can you unmute? Yep, that should be okay.
Okay. Yeah, I think it's good. Okay. Yeah, thanks very much, Yadvinder, for the introduction and for the invitation to speak in the seminar series. And yeah, looking forward to presenting some of the data that Yadvinder and his group have also been collaborating on. Okay, I think I'm ready to go here. I'll just make sure I've got a laser pointer. Great. So hopefully everyone can hear me well and can see the screen. If there is any issue at all, just let me know. Okay, so I want to talk about the ecophysiology of Australian tropical rainforest. This is a picture of one of the upland forests above the Daintree Rainforest Observatory, which is a research site. And here's a little bit of an outline of what I want to talk about. I want to first talk about some general considerations about tropical forest responses to global change, particularly rising carbon dioxide concentration and global warming. And then introduce a little bit about the context of Australian rainforests, because they are in many ways unique and different from, I mean, all tropical rainforests are unique, but there is a particular context for the Australian tropical rainforest. And then look at whether we can actually see some impacts of how warming and VPD might be shaping Australian tropical rainforest communities, and whether we can see any evidence for impacts of rising CO2. And to do that, I'll introduce a couple of case studies where we've gone into a bit more depth in terms of trying to understand the forest functioning. So I guess the sort of the larger scale I want to talk about is how rising CO2 is impacting plants. Of course, plants interact directly with CO2 through the process of photosynthesis, but as we're increasing the carbon dioxide concentration of the atmosphere, it's also leading to global warming. And so we often have, we can think of kind of opposing impacts on terrestrial plants of those two things. So CO2 is promoting photosynthesis because it's a substrate for photosynthesis. And on the other hand, warming and higher vapor pressure deficit can stress plants. And so those two things are, in a sense, especially in tropical rainforests, I think are opposing each other. And one of the most important questions then for predicting carbon cycle responses of tropical rainforest is to understand in which cases one or the other of those will predominate in its impacts. So I just want to then mention some couple of things as introduction and talk about two ways in which elevated CO2 influences plants. One is that for C3 plants, and trees are almost all C3, using the C3 photosynthetic pathway, a higher CO2 concentration will lead to a higher photosynthetic rate at the leaf level. And so this is something we observe experimentally and can demonstrate. You see here some nice data from Klaus Winter in Panama. And oftentimes we make these plots against the intercellular CO2 concentration. Here it's actually the atmospheric, the ambient CO2 concentration around the leaf, which is shown so that you can have some idea of how photosynthetic rates might increase. So at the moment we have a CO2 concentration in the atmosphere of about 400 parts per million. You can see that here. And you can see that there's still an increasing photosynthetic rate. And then it's a saturating function. So at some higher carbon dioxide concentration, the photosynthetic rate will no longer increase. So that's one very general thing about how CO2 impacts plants. And the other very general thing that we see experimentally and which has a strong theoretical basis is that there's an increase in water use efficiency. So water use efficiency can be expressed in different ways at the leaf level. We like to express it as the rate of CO2, the rate of carbon dioxide uptake by photosynthesis divided by the rate of water loss by transpiration. But we can also express it at the whole plant level as dry matter produced by a plant divided by the cumulative transpiration. And that's what's shown here. So this is for 10 different tropical tree seedlings. These are experiments that I did in Panama again with Klaus Winter. And here we watered these plants, kept track of how much water we gave them over the course of several months, and then measured how much dry matter they produced. And you can see there's this very consistent increase in the water use efficiency at elevated CO2. And we observe the same thing at the leaf level if we measure gas exchange with a portable photosynthesis system. Under elevated CO2, there's a higher CO2 fixation for a given amount of water loss. Okay, so then in a general sense, what are some of the ways that warming might oppose that increase in photosynthesis and the increased growth rate that we might expect from that? So one way is that higher temperatures are associated with higher respiration rates. And I think especially in earlier years and earlier decades, it was thought that global warming would lead to increasing respiration and impact upon the carbon balance of trees in such a way that the trees would have less carbon available to allocate to growth. They would allocate more to respiration. That would slow growth rates. And I think in the intervening years, we've learned that that respiration rates, there's a lot of acclimation potential. And here you can see some data from a warming experiment in boreal forest. So this is Peter Reich and his group's work. And they show that you do see quite a marked acclimation. So for plants that are grown at higher temperatures, they don't have the same high respiration rates as a plant which is grown at a lower temperature and then given a short-term increase in temperature. But this is nonetheless a really important and large, well-studied area of plant science. But I'm not gonna focus much on this any further than mentioning that. The second really important impact of a higher temperature is that it normally comes along with an increased vapor pressure deficit. And so how does vapor pressure deficit impact upon plant functioning? Well, one way, probably the main way, is that it can cause the stomata to close. Here you can see data from a canopy crane in Panama. Gerhard Zotz and Klaus Winter published these data. And we have kind of a typical diurnal course of weather conditions here. So here you can see the sunlight, the photosynthetic photon flux density. So in the middle of the day, there's more sun. And then you see this sudden depression of photosynthesis in the middle of the day. So A here is assimilation rate. So photosynthesis was very high in the morning, suddenly decreased. Why did it decrease? Well, because the stomata closed. So here you can see stomatal conductance has gone down to almost zero. And what are the weather conditions, the environmental conditions driving that? it's this increase in the vapor pressure deficit. So here you see vapor pressure deficit. So this is something that could potentially be really important for tropical forests because they're normally sitting at high temperatures, and a relatively small increase in temperature can lead to a relatively large increase in vapor pressure deficit. So let's look at that a little bit more closely in the next few slides here. So the term vapor pressure deficit refers to the difference between the amount of water vapor that the air could hold at saturation. That's represented here by this blue line. It's an exponential function of temperature, a well-characterized function. So this is how much water vapor the air can hold before the water vapor begins to condense. So that's the saturation vapor pressure. And the vapor pressure deficit is the difference between that quantity and the actual vapor pressure. And you can see that shown here as the difference between these two arrows. So what you can see here then is a constant relative humidity. This dashed line is a relative humidity of 20%. So if we hold the relative humidity constant at some value and then increase the temperature, we see a larger vapor pressure deficit at the higher temperature. Similarly, so this might be something kind of corresponding to a global warming scenario. That's one of the more straightforward ways to think about global warming, is that temperature will increase, relative humidity will probably remain about the same globally, and therefore the vapor pressure deficit will increase. So that's what would happen at constant relative humidity. Over the course of a diurnal cycle, we probably have something that's more like a constant actual vapor pressure. And so as the temperature goes up in the middle of the day, then you get a bigger vapor pressure deficit as well. So why is this important? Well, it turns out that in addition to warming of the land surface with global climate change, we also have increasing vapor pressure deficits. So this is a nice synthesis paper that came out last year. And so this is the globally averaged vapor pressure deficit anomaly. And you can see from the 80s or so, 70s or 80s, there's this pretty marked increase globally in the globally averaged vapor pressure deficit. So let's take a closer look at how that increasing VPD could impact upon leaf functioning. So here we have a kind of a cartoon drawing of the cross section of a leaf. The CO2, the carbon dioxide is diffusing into the leaf here. It'll be fixed in the chloroplast through the process of photosynthesis. And at the same time, the airspace inside the leaf is saturated with water vapor or close to saturated. We think most of the time. It might not always be the case, but that's what we often assume. And that water vapor then diffuses out through the same stomatal pore that the CO2 diffuses into. When the vapor pressure deficit goes up, more of the water vapor will diffuse out because the diffusion gradient gets steeper. And if the leaf then can't replace that water by water taken up by the roots as fast as it's lost, then the stomatal will close to slow the rate of water loss. And in doing that, it will also slow the rate of CO2 diffusion into the leaf. So at higher vapor pressure deficit, we expect to see both a lower photosynthetic rate and also a lower water use efficiency as well. These are some data for Poplar that I collected with colleagues a couple years ago in Switzerland, Greg Goldsmith, and who probably is familiar to many in Yadvinder's group, and Rolf Siegwolf. And it shows a really nice response of stomatal conductance to vapor pressure deficit. So here you can see this characteristic sort of decline. It follows a kind of an inverse relationship with vapor pressure deficit. And you can also see this nice decline, nicely revealed decline in the photosynthetic rate as well. So a linear decrease in photosynthesis with increasing vapor pressure deficit. this kind of encapsulates the sort of leaf level impact that we would expect to see. And I show these data. These are for poplar. It's not a tropical tree species, but the thing that's especially interesting and special about this data set is that we also had another poplar. So we had the wild type, and then also a transgenic that was insensitive to abscisic acid. And that insensitivity to abscisic acid meant that the stomata in the transgenic were essentially paralyzed. So you can see here images from a laser scanning microscope. And in order to put the leaf on the stage of the microscope, we had to detach it. So it was sort of detached for several minutes. And for the wild type, the stomata are closed, which is what we would expect kind of in those conditions. But you can see in the transgenic here, the stomata are still open, wide open, and so the leaf is continuing to lose water vapor. And as a result of that, you can see really nicely the job that the stomata are doing. So here's these are the same data again for photosynthesis, stomatal conductance here, and here's the transpiration rate, the rate of water loss. This is the ABA-insensitive transgenic, the one that doesn't close its stomata, sort of has paralyzed stomata. You can see a linear increase in the rate of water loss with increasing vapor pressure deficit. And it doesn't withstand very high vapor pressure deficit, about 2 kilopascals or so, before the transpiration rate starts to decline and it's no longer a steady state response. And so these data don't show that decline, they only show the steady state data. But nevertheless, you can see the job that the stomata are doing in the wild type. They're very nicely regulating the transpiration rate. And the consequence for the plant that doesn't close its stomata, you can see examples of the ABA-insensitive plant here in the wild type. The consequence is the leaves died. So they were exposed to vapor pressure deficits of about 2 kilopascals. And when we took the leaves out of the cuvette after making these gas exchange measurements, they were visibly dried like this, and within some short period of time, it was clear that they had died, that exposure to that VPD was fatal. So that's what the stomata are doing. They're protecting the leaf from desiccating, and we also envision that they protect the plant in such a way that it doesn't cavitate its xylem in the hydraulic pathway between the leaves and between the roots and the leaves. So I think one of the more challenging things that in terms of understanding the gas exchange of tropical forests over the last couple of decades is to try to separate is when we see a decrease in canopy photosynthesis associated with higher temperature, to try to separate the impact of the temperature per se from the increase in VPD. So the increase in VPD can cause the stomata to close somewhat, slow the photosynthesis rate and slow the uptake of carbon dioxide. On the other hand, the higher temperature itself could also impact photosynthesis and impact the biochemistry of photosynthesis and decrease the sort of photosynthetic capacity. So this has been a real challenge, and there was a paper published just recently that I thought had a really creative way of addressing this. And so I thought it's worth just taking a quick look. So these are data here from eddy covariance towers in the Amazon. And they show this covariance between increasing temperature and increasing VPD. So as the temperature goes up, there's a larger fraction of unsaturation, if you think of that graph that I showed earlier. And those two things are difficult to separate. The really creative thing they did here was to use data from the Biosphere 2 tropical rainforest. And here the increase in temperature is uncoupled from an increase in VPD. I imagine it's a result of the rate at which they can actually circulate air through that tropical rainforest enclosure. Probably means there's less mixing of the water vapor from the canopy airspace up into the atmosphere. And so as the temperature goes up, you don't see the same increase in vapor pressure deficit. So here they've plotted the canopy photosynthesis, this sort of gross ecosystem productivity, the carbon dioxide uptake by the canopy. And you can see, interestingly, as temperature goes up, then these responses diverge. So these are the data from the eddy covariance towers in the Amazon. These are the data from Biosphere 2 tropical forest. And you don't see a coherent response to temperature when you plot against temperature itself. On the other hand, if you make the plot against VPD, you do see this very coherent pattern for all of the forest canopies. And it suggests that it's actually VPD itself that's driving this, this reduction in canopy photosynthesis with increasing temperature. So I think that's a really nice insight, and it gives us some direction that we could take forward. This is another way that they also showed that. They binned the data, looked either at constant temperature at different VPDs or at constant VPDs at different temperatures. And again, so these are for the eddy covariance data. This again supports the idea that it's VPD that shows this nice coherent response when you hold temperature constant. And when you hold VPD constant and plot against temperature, you don't get such a response that makes sense in the same way. This is the title of the paper, Empirical Evidence for Resilience of Tropical Forest Photosynthesis in a Warmer World. So for me, this is a nice indicator that vapor pressure deficit is something that's important to consider in addition to temperature itself as well. Okay, so let's take a look at Australian rainforests. Of course, Australia is largely a very dry continent, the driest vegetated continent, and you can see the woody cover fraction here on the right-hand side from a nice paper that came out recently. You can see that the forest cover is mainly concentrated along the east coast here, Tasmania as well. And the tropics are kind of from here upward to the north. And we see this tropical rainforest here in northeast Queensland in this sort of thin stretch along the coast and up here on the Cape York Peninsula as well. So it's a very small fraction of the Australian land surface that's actually a tropical rainforest. It's about 0.3 or 0.4% of the land surface. But it's very interesting in terms of its history. So we know from fossil data that that Australia supported a widespread cool adapted rainforest for much of its history. It went through alternating periods of warmer, more tropical conditions and cooler, more temperate phases. Until about 38 million years ago, when Australia separated from Antarctica and began drifting north. And then there was, in the course of this aridification, a massive contraction of that rainforest to this tiny little refugia along the eastern coast and those ranges along the northeastern coast. There was interchange of flora, of plant species with lineages from Asia, as the Sahul plate that Australia sits on drifted north and came into contact with this Sunda shelf. And you can see here from about 20 million years ago or so, there were more exchanges, but the tropical rainforest flora has a Gondwanan component, and then it has this Laurasian component that's been mixed into it. Those Asian plant lineages have successfully colonized lowland areas, and then the upland areas, the sort of cooler upland areas, have retained a higher percentage of these Gondwanan lineages. This is the largest remaining rainforest refugium is in these upland areas, the Atherton, Bellin, and Kerr uplands. And this is it's a small area, geographically speaking. But it would seem the floristics would suggest that it's remained stable through time. serving as an arc for these relic lineages for over 40 million years. And if we just take a look at some of the statistics for the different plants, this area is referred to as the wet tropics bioregion. It contains over 4,000 plant species. 16 of the world's 28 lineages of primitive flowering plants are represented. It has very high endemicity. 31% of the bioregional rainforest flora is endemic, and much of Australia's plant biodiversity is conserved in the wet tropics. So it has high conservation value, and these really interesting floristics. And for me, when I think about these two different things, it gives two very different impressions. On the one hand, the Australian tropical rainforest looks extremely precarious. It's a little tiny sliver of the northeast coast. It looks fragile and vulnerable. On the other hand, it has the floristics suggest it's been extremely durable, resilient, and persisted for a really long time in geological time. And so when I think about climate change, I have these two very kind of different reactions. On the one hand, I feel very nervous. On the other hand, it seems like it's been through lots of climate changes. So this is just a little bit of a context, and I find something that's really interesting about and special about the Australian rainforest. Okay, so one of the nice tools that we have for learning, making insights into Australian tropical rainforest ecology has come from a series of rainforest plots that were set up by the CSIRO in the 1970s. You can see the distribution of the plots here in this region. So here's Cairns, where I'm based at James Cook University, and the plots are mainly concentrated around around this area. There's some up here on the Cape York Peninsula, and there's smaller patches of rainforest, and then one down here all the way to Mackay. So they're around about 25 plots or so, and they have quite a long time series of forest census starting in the 70s. So you can see here a bit more of a visualization. Another really interesting thing about the rainforest is that it's typically wetter on the coast, and as you go inland, you get to this really sharp boundary. So where it transitions from rainforest to savanna, and there's a particular community there of plants, the sort of wet sclerophyll community, which is really interesting in and of itself, and it's part of this sort of sharp transition from rainforest to savanna, associated with a rainfall gradient. So wetter near the coast and drier as you move inland. There's also a range of elevations. The tallest mountains in Queensland are about 1,600 meters. This is one of the Mount Bellinankura, and those mountaintops also support interesting and highly endemic plant communities. There are about 40 or so woody species which occur only above 1,000 meters. So these endemic mountaintop species, which have this particular distribution, which gives the impression that they are really vulnerable to global warming, because as the climate warms, the sort of climatic niche that they currently occupy will just get pushed up the mountain, and there's not very far for it to go. So the mountains are not tall enough that it can really migrate upslope for very far. Okay, so this is an analysis that David Bauman has worked on in Yadvinder's group, and he did a fantastic job of pulling together all of these plot data, along with some functional trait data that we collected, that I collected in collaboration with Yadvinder. And at the bottom, you can see the climate, the sort of interpolated climate for these different rainforest plots. And what you can see, of course, is an increase in temperature from 1970 through to 2020. And then you see these anomalies in temperature, the positive anomalies in red and the negative anomalies in blue, this overall increasing temperature trend. And along with that increasing temperature trend, also this increase in vapor pressure deficit. So this is, I think, a really interesting thing is to try to understand what impact the increasing vapor pressure deficit is having along with the temperature. So whereas they in the previous experiment that I showed that took advantage of Biosphere 2, where they could experimentally separate VPD from temperature, in this case, David was able to do that in a statistically using a hierarchical Bayesian analysis. So here you can see the effect sizes for the mean climate. So the mean temperature, we can see that as temperature increases, this is for tree growth rates. The growth rate is the response variable. So increasing temperature in terms of the mean climate at the plot leads to an increasing growth rate. That means so as you go from the lowland up to higher elevations, the growth rates slow down at the cooler temperatures at the higher elevations. And this we interpret as an impact of temperature on metabolic rates. So higher temperature means higher metabolic rates leading to higher growth. We don't see there's not a strong relationship in these data with the mean climatological water deficit in the soil. So this would be some combination of rainfall and evaporation, potential evaporation. And we see a positive impact of VPD. That's probably going along with that positive impact of temperature. More interesting is if we look at the anomalies in climate. And so here we can see a positive anomaly in temperature leads to a negative, has a negative impact on growth. So as we might expect, that's also associated with a higher VPD. So again, this VPD anomalies are having a negative impact on growth rates. And I think this provides some nice corroborating evidence to the other work that shows VPD having an impact on slowing photosynthesis and as a result, slowing growth rates. I want to then take a look at another kind of case study, which has been looking at the hydraulics of trees at two of these plots. So this is Robson Creek and Cow Bay. They're both sites that are more intensively studied that have eddy covariance and a range of other things being measured in addition to having the plot census data as well. So the plot at Robson Creek sits at about 700 meters, has a rainfall of plus or minus 2,000 millimeters or so per year. And the plot at Cow Bay is close to sea level and has about twice the rainfall or more compared to Robson Creek. So this is a project that's, that Patrick Muir is leading, and I'm collaborating on that. And then Oliver Binks, who will be familiar to many of you, I'm sure, is a postdoc that's leading the measurements. And so the data I'll present are data that Oliver has collected, and I think give some interesting kind of a consistent story with the data that the analysis that David Bauman did in terms of showing an impact of VPD. In this case, more so than the soil moisture, which was surprising, but perhaps is kind of coalescing around a common theme. So here are the climate data for these two different sites. Again, Cow Bay is at sea level, and Robson Creek about 700 meters. So this is the higher rainfall at Cow Bay, and then lower rainfall at Robson Creek. And the data here are shown next to each other, and Oliver has added these sort of boxes that can help your eye to pick out the differences between the two plots. So the interesting things to look at here, I think, are that the VPD is higher at Cow Bay because of the higher temperature, because it sits down near sea level, and at Robson Creek the VPD is lower. It's a little bit sunnier at Cow Bay than at Robson Creek, associated with cloud cover. And interestingly, here's the soil water potential. So as we expected with the lower rainfall, the soil in Robson Creek can reach a lower water potential in the dry season. So it has a drier soil and a lower VPD. Oliver was interested in looking at the hydraulic characteristics and the water relations of the trees. And we thought initially that this would be driven by the drier soil at Robson Creek compared to the wetter soil at Cow Bay. But we learned opposite to that, so the VPD trend going opposite to that. And it turns out they track more with VPD than soil water content in this case. So here, pre-dawn water potentials at the two sites. Cow Bay, so each of these two, the points on the x-axis is a species, a different species. And with no fill in the background is Cow Bay, and with the gray fill in the background is Robson Creek. And then these parts of the graph where you see it alternating gray and then white, gray, and then white are because there are some species in common. And so those have been aligned next to each other. So Cryptocarya macedoniana here is shown for Cow Bay in this white strip, and then for Robson Creek in the gray strip. And then we have our Rhododendron paralatum, Cow Bay, and Robson Creek. So it helps us to see, we can see the mean effects of the different sites. We can also see how species that co-occur at both sites are behaving differently between the two sites. The takeaway here is that the pre-dawn water potential is not different between the two sites. On the other hand, the midday water potential is. So and it's lowest at Cow Bay. So going along with the higher VPD at Cow Bay is a lower midday water potential and compared to Robson Creek. And then the real focus of the study was to understand the sort of hydraulic characteristics of these different trees. So this is the hydraulic vulnerability, the P50, the water potential at which a branch has lost 50% of its hydraulic conductivity. And we expected that, that this P50 would be lower at Robson Creek, but we thought it would be driven by the drier soils. In fact, it's lower at Cow Bay and corresponds to that higher vapor pressure deficit. So you can see the impact, the means for the two different sites, the blue line being lower for Cow Bay, suggesting that the vulnerability is less. So the trees at Cow Bay have constructed their xylem in such a way that it can withstand more negative leaf water potentials before losing hydraulic conductivity. And we can also see that it's the pattern isn't so clear for the different species where they co-occur at both sites, but it does tend to support the idea that that the same species has acclimated at Cow Bay to the higher vapor pressure deficit by having by constructing wood that's less vulnerable to cavitation. So it looks like VPD is an important driver of the ecophysiology of Australian tropical rainforest trees. VPD is increasing as temperatures increase with global warming, and I think this should be something that's important to understand how it will impact upon different species and upon the distribution of the rainforest as a whole. So what about rising carbon dioxide? So that's we expect to kind of go in the opposite direction to the rising VPD in terms of the leaf level effects. Can we see any evidence, what we would think would be evidence of impacts of a rising carbon dioxide concentration? The first place we might think to look is to look at the growth rates of trees on these forest census plots that have been around since the 70s, and to see if the biomass, the sort of plot level biomass, is increasing, as has been observed in other tropical forest plots over decades with increasing CO2 concentration. And what comes out of that analysis, I think for the most part, is that there's not really any detectable trend in biomass. And I asked David about if he'd looked at the growth rates, and I think that the result was sort of similar. Some species show increased growth rates, some species show a decreasing growth rate, and on average, it's probably not differing. With respect to the biomass, it's complicated by disturbances by cyclones. So in the footnote here, you can see it says the capital letter C refers to major cyclone disturbance of the plot. The lowercase c is minor cyclone disturbance of the plot. And you can see that there's a C, either a capital or a lowercase c, next to almost every single one of the plots. So this sort of disturbance regime associated with tropical cyclones is probably making increasing the variability and making it a bit harder to detect trends in biomass, decadal scale trends in forest biomass. Another thing, another place to look would be along this western edge of the extent of the rainforest. So this is the sort of drier edge where the rainforest gives way to savanna. And we could hypothesize that with increasing carbon dioxide concentration in the atmosphere, the water use efficiency of the trees along this drier edge would increase. And then we might expect to see the rainforest border pushing into the savanna. So David Ting did an analysis of aerial photographs over from the 50s and then into the early 2000s, and looked at 5 different locations, which you can see in these boxes here. So again, along this western edge of the rainforest distribution. And the result was that, so here you can see the proportion of change from rainforest to savanna. So some of the points that he looked at, there was the savanna had moved into the rainforest, or the rainforest had retreated. However, more often it was the case that the rainforest had expanded into the savanna. And so here's the net change in forest area. There's a net increase in the rainforest and at the expense of a decrease along this border, a decrease in the area of the savanna. So that would suggest, I mean, that I think is, we can try to hypothesize different drivers for that, but rising atmospheric carbon dioxide concentration would seem to be a reasonable guess at what's driving that. Okay, so I want to then take you to one other experiment. It's a drought experiment, so I'm going to talk a little bit about the drought, and then I want to try to bring that back, connect that back to carbon dioxide, to come back to that topic. So this is a drought experiment at the Daintree Rainforest Observatory, which is a research station run by James Cook University. It has a canopy crane, and Susan Lawrence has led the implementation of a drought experiment. You can see a picture of it here. It's a through-fall exclusion experiment, so the rain comes through the forest canopy, hits these panels, and then is directed into these gutters and directed away from the site. The experiment is set up below the canopy crane. There's a 1 hectare forest census plot with data that go back to about 2000. I think 2001 was the first census. And these panels cover about 0.4 hectares within that plot. So it's sort of, the plot is sort of split under the footprint of the crane between the drought experiment and a control side, so a drought side and a control side. Here you can see soil moisture data for the first 3 years of this drought experiment, and the panels are good at drying the shallower soil layers. You see that here. So the orange line is under the through-fall exclusion, and the green line is our soil moisture sensors on the control side. And you can see the wet season and the dry season, wet season and the dry season. So in the shallowest soil layer, we see the biggest difference. And then as you go to deeper soils, this is 0.5 meters depth. When you get down to 1 meter's depth and 1.5 meters depth, then the soil moisture is not very different between the drought and the control. It's probably partly a result of the fact that there's sort of lateral flow water from upslope down onto the drought plot and subsurface flow as well. So that's kind of the impact anyway that the panels have had. They've dried the shallower layers of the soil, but the deeper layers are not much affected. So these are then I'll show you some data for palms. They're actually a reasonably high number of palms. This black palm is the one that's shown here, Norman By. And the palm is interesting because it shows a really skewed distribution toward this creek on the north side of the plot. And for anyone that's listening that has visited the site, the track that you walk into the plot sort of comes in from this right-hand side, and then the crane is in the center of the plot. And so then over here toward the creek, you have this concentration of palms. So those are they're interesting because they give the impression immediately that they're sort of moisture sensitive, and that's consistent with the sort of general biology of palms as well. These are data that Nara Vogato, who's a PhD student with me, collected gas exchange data. We can see, in fact, that the stomatal conductance is lower in the droughted palms in the dry season. And this is the photosynthetic rates, and a little bit lower in the wet season. And here's the stomatal conductance. You see a seasonal impact lower in the dry season than in the wet season, and you see the droughted individuals, palms, having a lower conductance than the control side individuals. Also, that shows up in the carbon isotope ratio as well. So the carbon isotope ratio reflects the intercellular carbon dioxide concentration, which is related to water use efficiency. So we see a less negative delta 13C in the droughted palms, which is what we would expect given those observations of the gas exchange of the lower stomatal conductance. So that makes sense. I think that's all reasonable enough. We see an impact on the palms of that drought treatment. When we look at the tree community, we get a different result. So it's kind of surprising, but we've never seen a big gas exchange response in the other trees, and they aren't species that show this the sort of obvious skewed distribution toward the creek the way that the palms do. So I think there's good reason to think that the palms are shallow rooted and very moisture sensitive, and the rest of the trees. These are data by Alex Pivovarov, who's works a postdoc with Nate McDowell. They came as part of the Neotropics and did some measurement campaigns. And what you see here is that for the trees, this is 11 different species, only a single drought and a single control individual of each of those species, because it's hard to find enough replicates in the plot. But nevertheless, we see no impact on the leaf level gas exchange of the drought treatment. There does seem to be a shift in the wood anatomy, and this is also consistent with a previous paper by David Ting as well. So it seems like there's been acclimation in the trees toward deeper rooting, and probably some acclimation of the wood, the hydraulic characteristics of the wood as well. But nonetheless, they're definitely less sensitive than the palms. So the palms seem to be the most sensitive to moisture stress in this forest. And this is a reasonably wet forest. At this site, it gets about 4,000 millimeters of rain per year. So what could we expect from that in terms of CO2? Well, for me, this shows that even in a wet forest, the palms are sensitive to moisture. We might expect then that they might be especially sensitive to an increase in water use efficiency, which can come about from a higher CO2 concentration in the atmosphere. So is there any other reason to think that palms might respond more strongly to rising atmospheric CO2 than other taxa? This is a really interesting paper here by Carlos Jaramillo from the Smithsonian Tropical Research Institute, and it came out about 10 years ago. So he looked at soil sediment cores from Colombia and looked at pollen grains in different layers of those cores. And the cores went back many millions of years, and he could go back to the Paleocene-Eocene thermal maximum. So this was an event 56 million years ago where suddenly the atmospheric concentration of carbon dioxide increased rapidly in geological terms. So over about 10,000 years, went up to around 1,000 parts per million. Global temperature increased by about 6 degrees Celsius. And it's seen in a sense as a sort of some kind of analog for anthropogenic modern climate change, or at least something we could look to for some to maybe get some insight. So what did they find here? Really interestingly, after that event, that big pulse of CO2, carbon dioxide went into the air. There were some new taxa that showed up. There were some other taxa that declined. But what was one of the most impacted taxa were the palms, the Ericaceae. So suddenly there's a big diversification and increase in the abundance of the pollen of the palms, suggesting that palms could be especially sensitive to an increase in atmospheric carbon dioxide concentration. So can we look at the permanent plot network in the wet tropics to see if we might see something interesting happening with the palms? And this is really just the kind of crudest first approximation of an analysis that could be done. There are 4 of the plots that have a sort of reasonable number of palm stems within the forest census. There are a couple, maybe 3 or 4 other plots that have sort of 5 or 6 or 2 or 3 or 4 or 5 or 6 palm stems. I didn't show those, but here are the abundance, the change in the abundance for these plots, which are relatively rich in palms. And in these cases, it's a small number of plots, but there's an increase in the number of stems from the 1970s, the first census in the 70s through to the most recent census. And here you can see the percent of the number of stems of above 10 centimeters on the plot that are palms as a percentage of the total number of stems. And that increased here from 27% to 31%. So plus 4%, plus 15% here, plus 5%. And then at the Daintree Rainforest Observatory, so this is only for 17 years. The other ones are from the 70s. This one had its first census in 2001, but there's also an increase in palm abundance on that plot. So very anecdotal, but suggests that there could be something interesting happening in the species composition of these forests in the wet interior of the rainforest. And it could, I think, it could logically be connected to increasing atmospheric CO2 concentration. Okay, so just to wrap up then, I think we discussed some good evidence for the role that vapor pressure deficit plays in tropical rainforests in terms of the sort of leaf and canopy scale physiology. And I think that's something we should really take into account and consider in making predictions about forest function that can inform management or mitigation of human activities. I think there's a role for that rising CO2 is playing. And as I discussed in the beginning, these two are kind of competing with each other, the impact of rising CO2 and the impact of increasing vapor pressure deficit. It looks like that the tropical rainforest is increasing at least on the dry margins from those aerial photographs. I think it's important to try to look closer, even though there's this high incidence of disturbance associated with cyclones, to try to understand what's happening in the interior as well. And then something I didn't talk about very much, but I mentioned is these mountaintop endemic species. We really have no idea if they're restricted to the mountaintops, to the higher elevations above 1,000 meters because of temperature in and of itself, or because of other ecological interactions or soil conditions. And they are, I think, if it is temperature that's that restricts them, then they're really vulnerable to extinction, because yeah, their temperature, their climatic niche with respect to temperature will soon disappear. So I think that's also something that we should really focus on. We part of a project at the moment with Darren Crane and other colleagues in the Tropical Herbarium, where they're focusing on collecting these species for ex situ conservation, and we're also making some physiological analyses as well. So hopefully we'll learn more about that. Okay, thanks. Thank you very much for listening. And if anyone has any questions, I'd be more than happy to discuss.
Okay, thank you, Lucas. I'll give you some applause. That's all best you can. So if you stop sharing your screen, we can open it up to the audience for questions. I encourage you to switch on your cameras. It's quite nice to see a physical audience, or a visual audience at least, in these things. If you can, do switch on your camera and microphone. Even if you're just listening in. And if you ask a question, it'd be nice to have your camera on. So either we don't have any questions lined up in the chat, so I encourage you to pose a quick question or a short note on your question in the chat, and then I can share that and ask Lucas those questions. And I'll answer the first question from Livia there. Yes, this has been recording, so it'll be shared on my YouTube channel in a few hours' time, and I'll send I'll post it there by the end of today in Oxford time, and all the previous talks are there as well. Okay, while we're waiting for questions, do feel free to post some questions. I'll start off with one or two, Lucas. One actually is a general biogeographical question. You spoke of your concern about these little pockets of Australian forest that are there on the mountains. And it made me wonder, because Australia, through much of its history, has been connected to New Guinea. And how much similarity is there between the structure? Is New Zealand really just the extension of this Australian forest biome in terms of its composition, that just happens to be cut by a bit of water at the moment? Or are they quite distinct?
Yeah, I think that's a very good question. I think that there should be quite a lot of similarity, I would think, or at least there was certainly a land, connection in the last, before the Holocene and last ice age when sea level was lower. Yeah, excellent question. I don't actually know the answer, but I'm going to ask my colleagues in the herbarium and find that out.
Okay. If anybody else has the answer, feel free to answer it, because a question that just occurred to me when I saw your map. Okay, on to a more, perhaps a question more in your comfort zone. So the story you're telling with the palms, if I understood it correctly, is that, I don't know if it's just about palms or it applies to the dicots as well, the water sensitive species may be the ones that do the best in the wet rainforest, because the benefit they get from higher CO2 outweighs the costs of higher VPD and the negative elements there. So that's why you were suggesting that palms could be proliferating. That seems to be a different message that say stuff like Adrian Esquivel and Mulbert's work with in the Amazon with the plot inventories has been suggesting, where there's arguments that species that have drier ranges seem to be doing better in the Amazon rainforest than the wet sensitive species overall. So being restrained to a wet niche is probably makes you more vulnerable in this changing world. And I wonder if you have any thoughts on that. Maybe the nature of change in the Amazon, where you've got stronger droughts and dry events coming in, maybe is a difference from Australia. But I don't know if you have any thoughts on it.
Yeah. I guess my thought would be that that these particular forests are pretty wet, and like for example at the Daintree Rainforest Observatory, yeah, it's getting about 4,500 millimeters of rain per year, and so my feeling is that the palms are kind of shallow rooted, that they, because of their particular biology, they don't have secondary wood formation, and so I think the stomata should be especially sensitive to kind of protecting the the hydraulic architecture from embolism. So they might have an especially sensitive stomatal response to soil moisture deficit. And yeah, and interestingly, I think that they, because of that moisture sensitivity, could benefit from higher CO2. But I guess it might well be restricted to these. Well, they themselves are restricted to the sort of really wet sites and sort of wet site specialists. So I can also easily imagine that that species that are more dry forest, dry site specialists would benefit from an increase in water use efficiency as well. I think that would make sense. Yeah, I think, yeah, in terms of finding a fingerprint, ideally we might find the most sensitive species and sort of look more at that.
Mm-hmm. So just to follow up on that, do you think it's something peculiar about palms and their architecture and physiology, or is it?
I think, yes, I think so. I think that they, at least that makes sense to me, that they, because of their inability to replace damaged xylem, that they should be particularly conservative, and that might they might especially benefit from higher CO2.
Mm-hmm. Okay, I haven't so. Okay, we have a question from Vivek. Would you like to ask your question directly?
Yeah. My question is, hi Lucas, I'm from India. I'm right now working on soil fertility issues in remote district of Odisha state in India, where it is subhumid climate, and it is human dominated landscape where you could see increasingly with introduction of intensive agricultural practices among Aboriginal farmers who had been practicing shifting cultivation. Now the tree cover density has been decreasing gradually since last 4 decades. I don't know how to understand, your presentation was very insightful. How we could understand VPD dynamics play its role in subhumid landscapes where we could see extreme differences in seasons, like during winter and autumn the climate is very cold. During summer it is severe heat, and we can't see even grass cover on the soil. So with this kind of dynamics, how it will impact soil fertility, whether VPD dynamics could inform anything for this region. Sorry if it is appropriate.
Yeah, thanks, Vivek. Yeah, I think that's a really interesting question. For me, the interesting thing about the VPD is it should be kind of gradually increasing with global warming as the temperature increases, and so it could be this sort of pervasive thing that, you know, sort of increasing in the background, and then on top of that, then you'd have climate anomalies and seasonality, and you know, could have regionally droughts or more rainfall or less rainfall. So I think it would certainly interact, and in the case where the soil is also drying, and it's typically the case with a drought that the temperature at that particular time of year is higher, and so the VPD is higher as well. And then under a higher VPD, it will probably, you know, drive the trees to a negative response to the soil water deficit faster than they would have otherwise experienced, I think.
Yeah. Thank you very much. Yeah.
Okay, thank you. Any more questions? Feel free to just pose them directly, as in there's nothing queued up in the chat box. Anybody has a question, feel free to.
Yeah, I have a question. So, Lucas, so you presented a lot of good data on the impact of vapor pressure deficit, and you showed data for at least one of the sites with an increase in vapor pressure deficit over a few decades. So I'm wondering how universally accepted is it that vapor pressure deficit will increase with warming? And I'm particularly trying to make sense out of these papers that are out on this pan evaporation paradox that show that with increasing temperatures over the recent decades, you don't have an increase in evaporation from, sort of meteorological pan measurements of evaporation. And in fact, often they see a decrease, which suggests that vapor pressure deficit has been decreasing. And I don't know, have you looked into this and at all?
Yeah, that's a great question, Bill. Thanks. Yeah. So I think I remember asking Mike Roderick about this, and his response was that he thought that the pan evaporation seemed to be a really good metric for sunlight, and that the decreasing pan evaporation could, I think that was one of the ideas, that there was sort of increasing diffuse fraction of diffuse light fraction associated with sort of aerosols or some other atmospheric conditions or pollution. And so I guess but I think with warming, with global warming, the most kind of common prediction seems to be a more or less a constant relative humidity, which would lead to the higher vapor pressure deficit at a higher temperature. So I think that that's still a valid kind of expectation. Yeah, I think the stuff with pan evaporation is really interesting, and really something we need to work hard to understand. I definitely agree with that.
Just to pitch in on that as well, it isn't just a prediction. Just like you showed with David Bauman's data from Australia, there's an analysis from Australia. There's many decades now of observation that seem to consistently be showing increasing VPD over time. And some of these are from global products, so you have to look at what's built into the assumptions there. But I think overall there is quite strong evidence of relative humidity not increasing while temperature has been increasing. Okay, any more questions? So yeah, so while we're waiting on other questions, it is interesting that you come down to show this strong impact of VPD, and I find that in a number of studies that wherever we look, when we look at tree growth rates and their variation or other factors, we always find VPD as this strong predictive variable, much stronger than temperature, usually, and stronger often than soil moisture conditions as well in predicting seasonal or interannual variation in productivity or growth. And it seems like it's an insight that hasn't really extended much beyond the ecophysiological community. Now, when people think about the impacts of climate change on vegetation, often it's about drought, rainfall drought, or it's about temperature sometimes. But the fact that the atmosphere is becoming drier and sucking more moisture out of the biosphere as a phenomenon is something that somehow I feel hasn't reached outside the specialist science enough as a concept into sort of wider scientific or public awareness. It's not really a question, but it's just I
agree with that too. It's in some sense maybe just a little bit harder to grasp than drought or but I do definitely agree with that.
And what the study suggests, and I think what a lot of our evidence is suggesting now is that that's maybe really stressing vegetation over the coming decades is the increasing VPD, not the temperature per se. And so
and I guess rainfall is hard to predict, but uncertain. But that the increasing VPD is more. I think is more clear.
Okay, well, I'll pitch with a final question. I think unless anybody has one, we can finish after that. I'm just wondering, applying these insights into the past, when we think of the again this appreciation that's this interaction between VPD and CO2, who wins out of that seems to be determining productivity and also potentially species abundances and competition. And if you look back at the ice ages, lower CO2 and lower temperatures. Are there any insights from what won and lost in the colder conditions that can tell us about the relative importance of CO2 versus VPD in influencing growth? You drew some insights there from the Eocene maximum, but I wonder what past.
Yeah, that's a really good question. There are sort of pollen data from in the wet tropics. I must look into that and see. Yeah, very good question. I think I don't know, but I think that there would be some data that could be brought to bear that would be worth looking at. Yeah.
It's a bit similar, but I just started thinking that would it be possible to reconstruct like historical VPDs the same way that we can kind of reconstruct old temperature series? But it's completely something I completely don't know anything about, but like thought.
Yeah. Are there proxies for?
Yeah. What would you need for that?
Well, I guess your water use efficiency could help in the isotopes that give you the water use efficiency.
Yeah, that's right. Yeah, we certainly assume that the CI on CA, which we could get from carbon isotope ratio, is decreases as VPD increases, and so that would that could provide some means to try to reconstruct it. Yeah, from tree rings, I guess, and possibly from the delta 18O of the tree rings is also, although they're kind of number of other parameters that might change, but but I think that's that's something that people have been working toward in terms of the oxygen isotopes, that you could use that in conjunction with the carbon isotopes and get some information about VPD as well.
Mm-hmm. Oh, there's a possible new research direction there. Okay,
I have
go ahead.
One question. Yeah, I'm working about here smallholder farmers decision making team. So my question is, what is the current state of ecophysiology research community? Where is ecophysiology research is happening in human dominated patchy forest parcels also, or it is only happening in remote forest patches at present? Globally, what is the trend with the ecophysiology community, or the ecophysiology community's insights are now is at the stage whether we could readily use it for informing the human decision making among farmers, especially? How could they ecologically engineer their landscapes to increase the overall well-being? Yeah, thank you.
Yeah, I think that there's certainly an kind of effort toward being able to use traits, kind of functional traits and species traits in forest restoration to choose species that would be more suitable for a given landscape or a given climate, and perhaps thinking about future climate as well. And so I think that's, yeah, I think there's a reasonable convergence of ecophysiological research with restoration, with kind of revegetation and restoration in that sense. So others might have other insights as well.
Yeah. Okay. Thank you.
Okay. If there's no more questions, I guess it's time to thank you, Lucas. Thank you not only for giving us insights into Australia's rainforest, but I think a very lovely, clear exposition of just explaining gas exchange and the playoff between conductance and gas exchange in a very clear way. So it's fantastic to show that as well. So thanks for that, and good luck with the season and any work you're doing out there.
Thanks. Yeah. Thank you to everyone as well in the audience. Really a nice opportunity to present to you.
Okay. So bye-bye.